To understand the human mind, we must discard the metaphor of the brain as a rigid computer and look directly at its biological reality. This exploration begins with established science. We are not going to invent any new biological mechanisms to make the math work. Every physiological rule we examine is already widely accepted within contemporary neuroscience. Our immediate goal is to strictly define the physical constraints of the nervous system as they are understood today. Mapping this hardware is an absolute prerequisite. Before we can evaluate any theoretical model of higher cognition, we need to know exactly what the underlying biological machinery is actually capable of doing. We will build this map sequentially, starting down at the microscopic behavior of individual cells and working our way up to the macroscopic development of the entire brain. By structuring the review this way, we can identify the specific, recurring organizational principles that dictate how the nervous system functions at every scale. Cognitive theory must align with empirical biological reality to remain valid. Without that foundation, any model of the mind remains speculative. The starting point for all neural computation is the electrical properties of the individual neuron. Activity here is driven by a constant pull-pull dynamic. Excitation attempts to drive the cell toward action, while inhibition suppresses it. The immediate outcome determines whether a specific neural population breaches a critical threshold and fires an action potential, or remains silent. But this electrical hardware physically alters its structure based on experience. Through Hebbian plasticity, when neurons fire together, their connection is permanently reinforced. Mechanisms like long-term potentiation strengthen used pathways, while long-term depression prunes neglected ones. The fundamental unit of the brain is a continually adapting biological relay, permanently shaped by its own activation history. If we pull back from that single synapse, we can observe how sensory information moves across massive tracts of the cerebral cortex. What we see is a distinctly hierarchical architecture for processing data. Raw environmental features enter at the lowest levels and are progressively transformed into increasingly complex representations as they ascend the network. Historically, some suggested these complex ideas eventually land in a single, highly specialized cell. That is biologically inaccurate. Modern neuroscience demonstrates that representation relies entirely on population coding. A single concept, object, or input is encoded as a widely distributed pattern of simultaneous electrical activity rippling across vast neural networks. Distributing the workload this way makes the system robust, protecting our memories and skills from localized damage or single-point failures. Perceptions and memories do not occupy specific physical coordinates in your brain. They are orchestrated states of network activity. To see how these states sustain themselves, we transition from the spatial map and look at how the network behaves over time. Information doesn't just travel forward. Through recurrent dynamics, data loops back on itself. These feedback loops allow neural activity to stabilize and persist, maintaining a representation after the trigger disappears. This persistent circulating activity biases how the network processes the next incoming signal. Crucially, these internal dynamics are physically bound to your bodily actions. Through continuous sensory-motor interaction, your active physical exploration of the environment constantly alters the sensory inputs feeding back into your nervous system. Perception emerges through a continuous, physical feedback loop between an organism's movements and its surroundings. Zooming out to a whole brain perspective, we see how these disparate sensory, memory, and motor regions synchronize into a single functioning entity. Association cortices physically bridge the gaps between networks, integrating distinct streams of data across language centers, memory storage, and executive control. This integration is managed by widespread neuromodulatory systems, utilizing chemicals like dopamine and noradrenaline. These systems do not encode specific pieces of information themselves. Instead, neuromodulators act as a global gain control, regulating the excitability of distributed circuits without altering physical wiring. Your motivation and attention dictate these chemical shifts, priming entire networks for action or diminishing them. The final synchronization component is neural oscillations. These rhythmic, synchronized waves coordinate precise communication between vastly separated neural populations, aligning their firing patterns across multiple scales of time. The nervous system is a globally coordinated entity, governed by shifting chemical states and precise temporal rhythms. If we apply all of these mechanics across a complete lifespan, from embryonic development to old age, a clear evolutionary engine emerges. Early life generates chaotic, exuberant neural connectivity. Over decades of adaptation and expertise, that excess is systematically pruned back to create efficient, stabilized pathways. We have mapped a system that is distributed, hierarchical, recurrent, actively engaged with the world, and continuously adapting. One recurring organizational principle drives all of these seemingly diverse mechanisms. That principle is competition. At the microscopic level, excitation constantly fights inhibition to control cellular firing. That exact competitive dynamic scales up the hierarchy. Motor programs compete for physical execution, memory processes compete for conscious attention, and developmental plasticity stabilizes winning networks while pruning the losers. Competition is the primary evolutionary engine organizing the entire nervous system across every single spatial and temporal scale. We have now established the empirical baseline. We have a robust, complete understanding of the diverse, competitive mechanisms interacting within the physical tissue of the brain, which brings us to the core, unanswered question. Can the continuous interaction of these low-level biological mechanisms collectively give rise to a broader, unified computational principle? While these biological mechanisms explain how signals move, they do not yet explain how those signals become subjective thoughts or complex reasoning. Closing that gap requires an explanatory framework capable of integrating these disparate findings. The following proposal attempts to integrate these findings into a unified account of perception, cognition, and adaptive behavior. We call it tokenization theory. This theory serves as a conceptual bridge, connecting empirical neurobiology to the specific computational requirements of the human mind.